Photoluminescence scanning. While trying to get a Raman scan on a stone, it might get a reading which essentially doesn't show Raman peaks at the fingerprint area, but some emission peaks in other parts of the spectrum, like in this case. This happens because the stone contains elements which trigger photoluminescence reactions once exposed to the laser beam. This emission can sometimes be strong enough to make a Raman signal impossible to be detected by the spectrometer. Chromium is the most notable example of an element causing a strong PL reaction. This chromophore element is common in stones like ruby, sapphire, emerald, spinel, alexandrite, and many others. Even a minimum quantity of chromium can trigger a PL reaction that can make the Raman signal practically impossible to be seen by the spectrometer. The GEMO-Raman can even work as a PL spectrometer in the extended range, and so it's possible to identify stones containing chromium by specific PL emission peaks when it's not possible to get a Raman spectrum. While Raman peaks are measured in cm-1 on a relative scale having the wavelength of the laser, in our case 532 nanometers, as a zero value, PL peaks are always referred to an absolute scale expressed in nanometers. Since the software will feature both spectra on the same screen, for practical reasons the Raman scale is displayed at the bottom, while the nanometer scale where to read the PL peaks values is on top. Our task is to identify this blue stone, but we are not sure if it's better to proceed with the Raman or PL spectrum, so we will try with the pre-scan first. Click on Raman. It seems the stone had a huge reaction to the laser beam and produced an emission far from the Raman fingerprint zone. The resulting Raman spectrum seems to have few peaks riding above a broad slope caused by this strong emission, and it turns to be useless for the identification of the stone. So we will click on Pre-Scan again and choose the PL option this time. The software will now consider the whole spectrum and will calculate the best exposure time and averages for having all the PL peaks displayed. The operator has now two choices: to launch the autoscan or continue manually by running the dark and full scan. We will choose to click Dark and Full, because despite the autoscan feature works exactly in the same way for both Raman and PL spectra acquisition, it's usually not recommended for stones exhibiting strong PL features. The reason is that while a Raman spectrum is always baseline corrected, a PL spectrum is not. The baseline correction removes any slope from the base of the spectrum caused by broader photoluminescence bands. This allows for more precise spectral matching against the Raman library. Obviously, nobody wants to remove PL-generated slopes from the spectrum when focusing to study PL reaction. Thus, broad-range PL spectra are not baseline corrected. This is even the reason why the PL database is labeled as experimental. The extent, size, and course of the PL emission spectra can be so variable and different from each other that it's practically impossible to develop an algorithm allowing for consistent automated search. Once recorded a PL spectrum and activated the search in the PL database, the operator will have to visually check the results from the menu in order to be sure about the consistency of the matching reference provided by the software. Chromium and corundum produce the same PL peaks regardless of the color of the stone, so it's not a surprise we are getting ruby as a first matching reference, even if the stone is actually blue sapphire, which is displayed as a second choice by the automated search. Notice the other stones listed have a completely different PL spectra. There are instances where the concentration of the element causing the PL emission is so high that the spectrometer cannot see further light at specific wavelengths and gets blinded, saturated, or overexposed. This is similar to what happens to digital cameras where too much light can burn the image so that only white can be seen. If a CCD pixel gets overexposed, no further photons can be detected by this specific pixel during the ongoing exposure period. The software warns about saturation during the scan process by showing the following red message on the spectrum graph. Note: spectrum is partially saturated. The resulting spectrum will display a red portion where the saturation occurred. This issue can be solved by using the neutral density filter included in the accessories of the Gemo Raman. Open the sample chamber and place the filter on top of the sampling disc. Put the stone on the center of the filter and close the lid. Click Prescan, then PL Spectrum, and then Darken Full. The filter reduces both the intensity of the laser and the PL emission of the stone, allowing for the spectrum to be correctly recorded and displayed. It's important to underline that since the laser beam is focused on the surface of the sampling disc, the Raman fingerprint spectrum taken with the neutral density filter cannot be used to identify a stone placed upon it.
Thank you for watching this video. For more information about our spectrometers, please visit our website. And if you have any question, please don't hesitate to contact us at info@gemoraman.com.